Okay, so while people are joining, I'm just going to give a quick overview of the webinar today. We are going to be discussing bipolar disorder, which I'm sure many people who are joining have some familiarity with, but some people might not, or might not be familiar with what medical science knows about bipolar disorder. And then we will hear from all of our panelists about their experience in both the clinical and the research side. And then we'll bring everybody back on screen to discuss their collaborations and how they're working together to hopefully make life a little bit better for people that suffer from bipolar disorder. So we're going to address questions at the very end. Please feel free to put your questions into the Q&A feature of the webinar, and we will do our best to answer them live at the end. But if there are any outstanding questions that we don't have time to answer, we will try to collect them and email them out to everybody who joined, so hope that those get answered. And I'm going to start by introducing all of our panelists. I know that they were in the email confirmation that everybody got, but I would like to start by doing that in person. Joining us today, we have Andy Nierenberg, who is the director of the Doughton Family Center for Bipolar Treatment Innovation at Massachusetts General Hospital. Welcome, Andy. We also have Jenny Tam, who is a principal scientist and the leader of the synthetic biology platform here at the Wyss Institute at Harvard University. And we have Chandra Williams, who has a master's of education and is an expert bipolar researcher by experience, who's joining us all the way from California today through the magic of the internet. My name is Lindsay. I will be your host for today. I'm a science writer at the Wyss Institute for Biologically Inspired Engineering at Harvard, which is sponsoring this webinar. And I think we're going to go ahead and get started. So we're going to spotlight Andy for now, and Andy and I are going to talk about bipolar disorder from a clinical perspective, because Andy, your career has focused on bipolar disorder for a long time. You've seen a lot of patients. So can you give us an overview for anybody that might be joining that doesn't have direct experience? What is bipolar disorder? What does it do to patients, and what do we know about it?
So bipolar disorder is a disorder where people have changes in their mood and energy, and that this can be a very dynamic process. Sometimes people can have ups and downs, and I'll explain those in a little more detail, where it just seems to come and go, and that it can interfere with their lives in substantial ways. And in some ways, it can also enhance their lives when they are full of energy and can be very productive. But it's heterogeneous. It's very different for different people. Different people have different courses over time. And they can also have other things that go along with bipolar disorder, like anxiety, alcohol use, substance use, and some other things that make it even more difficult.
And you mentioned, Andy, that there are sort of highs and lows, and you mentioned anxiety. So how is bipolar disorder different from somebody that might have a diagnosis of just anxiety or just depression?
So anxiety is, that can wax and wane, but is fairly constant, which is characterized nervous, keyed up, on edge, worrying excessively. With bipolar disorder, when people have episodes, they can become manic. And when people become manic, they are bursting with energy to the point that they're thinking too fast. They even can't keep track of their own thoughts. It can be manifest by people talking too fast, and other people can't keep track of what they're saying. They need less sleep. They can think that they're thinking sharper and clearer, and sometimes that's true, but it can get to the point where it can interfere with their ability to link one thought after the other. The other thing that can happen is that they can lose their inhibitions and end up doing things that they later would regret, or when they weren't manic, they would never do anything like that. Driving too fast, sexual indiscretions, spending their life savings in a day on things that would be frivolous and they wouldn't ordinarily do.
And then when they would sort of come down from that, I guess, does it feel to them all of a sudden like someone else took control of them and did these things that they would never do? So does it feel kind of like a not a Jekyll and Hyde situation, but is it kind of, they're aware of what they're doing, but they don't feel like they have control over it?
It's more, many people will feel regret that they had done this. And once people actually understand that they're going through this, they'll recognize it afterwards and really feel pretty badly about it and try to clean up the mess that they've made for themselves, which is difficult for them to do. But when, people can get manic, they can get a little less manic, called hypomania, which is of a shorter duration and not as severe. They can feel okay. They can have residual symptoms, and they can get depressed. And when they get depressed, it looks like any sort of major depressive episode where people have low mood, low energy, can either sleep too little because of insomnia, which is different than mania. They need less sleep, or they could sleep too much. They can eat too little. They can eat too much. They can have a lot of self-negative thoughts and blame themselves for all sorts of things, have self-loathing, can feel guilty, can actually even move slower than they normally would, but they can also be agitated, and they can have suicidal thoughts and behaviors because they just feel horrible and can't stand it.
That sounds awful. I'm just gonna
It's
say it out loud.
it's
It sounds like
very painful for people to go through these states. And by the way, when people are manic, they can sometimes get euphoric, but they can also get irritable. And sometimes it's all mixed together, where people can, believe it or not, they can be manic and depressed at the same time.
Wow, that just sounds really difficult to parse. And especially if this happens to someone, repeatedly over and over, swinging between these different states. I understand why you say it's disruptive.
Yeah.
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 So what is available for patients right now? How is this treated? Can treatments help? How well do they work? What kind of options do people have right now?
So the treatments will depend on what what state, what episode somebody is in. So there are a whole set of treatments for mania. There are somewhat separate treatments for depression. And many times people have to take, on average, about 3 medications to try to be as stable as they can. There are some people who are very lucky and they respond to one of the mood stabilizers, the classic one being lithium. And maybe about a third of people can do well with lithium over the short run and over the long run. But a lot of times people will need the other medications that are indicated, for example, for mania. And those tend to be what's called dopamine blocking agents. Also, the class is the antipsychotics, which is unfortunate because people don't necessarily psychotic. Although I did leave out that people can have hallucinations and delusions when manic or depressed. So that's another aspect of what can happen. So you have a bunch of medications that are available for mania. You have limited medications that are available for the depression. The whole area of whether the classic antidepressants will help is controversial at best, and the data are weak at best that they can actually help. And there are some other treatments which I can tell you about later if that's relevant.
Okay, so it's interesting. You said that only a third of patients seem to be helped by sort of the frontline treatment for bipolar, which is not a great success rate.
Well, in terms of all of medicine, it's not that bad. In all of medicine, things don't work as well as one might think. But there's a big area in the field trying to understand what is it about the people who respond to lithium that's different from the people who don't respond to lithium. Because we really don't quite understand how lithium works, even though it's been around since 1948.
I think lithium is one of those drugs like aspirin, where
Exactly, right.
we still don't really know how it works. All we know is that it works. And so we keep prescribing it. And, if it works, great.
Right, right.
So what do we know about the underlying causes of bipolar? Because I know that with depression, sort of one of the predominating theories is that there is an imbalance in the amount of serotonin that's released in the brain. And so you have selective SSRI, select serotonin receptor inhibitor drugs. I know I'm butchering that. But is there any kind of a sense of biologically how this is different from depression? And are there any drugs that are specifically being developed to treat those underlying causes?
So we have a lot of information about the systems in the brain that are dysregulated. So it's not so much of an imbalance. It's more of a dynamic dysregulation of a very, very complex system. So we don't really know what's the fundamental cause of depression, and we don't know what the fundamental cause of bipolar disorder is. We do know that it's highly heritable. And there's been a very big push to try to identify the genetic architecture and what may be different in people who have bipolar disorder compared to people who don't. It's very, very slow progress. And although in the past month or so, there's some interesting findings that are suggestive of what might be involved. But one of the things that Chandra and I have talked about is that we're starting to reconceptualize bipolar disorder not as a mood disorder per se, but as an energy dysregulation disorder. And that there are very complicated ways that our brain uses energy that can be easily dysregulated.
Got it. And I have one last question for you, Andy, which is, in an earlier conversation, you had said that even though so many people have bipolar, it's actually a very prevalent disorder. You said that it can also almost be considered an orphan disease because there are so few treatments available and there's such a big unmet need. So why do you think that is? Why do you think that despite so many people having it, it hasn't really had a lot of progress?
Well, one thing is, look, it's a brain disorder. And you could say the same thing about ALS, Huntington's, and Parkinson's, right? That you know, the outcomes are not great for those brain disorders where we know the genes, we know exactly what's going on, we know the physiology, and yet progress in medications is slow. And again, I think that's because we tend to underestimate how complex the brain is and how difficult it is to re-regulate things that are dysregulated. That's one thing. The other thing is that, you know, there's been a lot more attention and funding put into schizophrenia and into depression. What's odd and hard to understand is that bipolar disorder is 2 to 3 times more prevalent than schizophrenia, but somehow schizophrenia is seen as more severe, but it's really not. I mean, these are really severe disorders that impact many millions of people. And at a minimum, you know, it's 2% of the population. You're talking about 6 to 7 million people in the United States alone who you know, have unequivocal, clear diagnoses. There's another 2% where it's a kind of spectrum. So it's a lot of people, and you know, we really need to have better understanding of the pathophysiology and better drug development.
Awesome. Well, I think that you've really encapsulated the unmet need there, which is great. Thanks, Andy. I'm going to let you go for a minute, and we're going to spotlight Chandra, who again is joining us from California. And Chandra is an expert researcher in bipolar disorder by experience. So you have some firsthand experience with some of the things that Andy was describing. So I would like to just open by asking you if you could just tell us your story about your experience with bipolar, to whatever detail you feel comfortable.
So I'll start by saying I was diagnosed with bipolar in July of 2015. Prior to that, in 2005, I received a diagnosis for anxiety, depression. And so just forwarding to, I'll say 2014, life was like amazing for me. And all I knew was that I would sleep a lot. I would sleep a lot because when I would go to sleep at night during the night hours, I would wake up 10 to 12 times a night. So it would make me have to sleep longer periods because I wasn't getting restful sleep. So that was the main symptom, and my anxiety just didn't leave, whether life was good or not. And so I knew I had to look into it. And so in 2014, I had a really stressful event that happened. And usually with me and stress, the insomnia comes. Okay, so the insomnia started probably October of 2014, and I just wasn't sleeping. I wasn't sleeping, and so by January 2015, the insomnia turned into mania. You know, because after a while, with the brain not sleeping, you know, that's what happened. And so I actually went into full-blown psychosis at 36 years old, you know. And when I came to my right mind again, I was in an institution. So I spent 5 days in an institution. And during my time in the institution, they're like, we don't know what it is. We don't know what's wrong. So they're like, you know what, you got to check it out on your own afterwards. And so I ended up getting pregnant shortly after my psychosis, you know, because as Andy said, you lose your inhibitions, you lose sense of time. And so I had to find a specific doctor, and I ended up being diagnosed out of a maternal fetal medicine program with USC. I was given the diagnosis of bipolar with psychotic features. But my doctor told me, they said, we're going to put you on this medication, which is Seroquel. And if you take your medication, you most likely won't go into psychosis again. So that was in July 2015. And so from then to now, what I've done is track my symptoms, because when I was told that I had bipolar, I was told that it was a mental emotional disorder. Some days you're happy, some days you're sad. That's all I had to go with. And so when I looked at the symptoms that I was experiencing, the anxiety, the fatigue, low muscle energy, meaning my muscles are toned, but there's something about the muscle energy where I'm not able to really lift anything. I'm not able to do anything that requires muscle energy. And so just taking a look at the symptoms, I said, you know what, these symptoms are more of physiological symptoms and not what people call a mood disorder. And so that's what I've done. I've spent my time tracking my symptoms, for me, in hopes of, I think my ultimate hope in tracking my symptoms is to create a symptom tracker specifically for bipolar. Because I think I received the depression scale and anxiety scale. But because bipolar depression and anxiety is different, there needs to be a different type of tracker, you know? So that's one of the things that I hope to influence, you know? And so, yeah, bipolar is very tough, because even now you wouldn't know, but I'm in a type of bipolar depression. When we say depression, we mean like fatigue. It's a type of fatigue that you get when you're sick, when you're fighting like a sickness. And it is very, very challenging to live with. I would say like the psychosis and the hallucinations, outside of me taking Seroquel, if I didn't take Seroquel, you know, because I've been in psychosis once. It's something that my brain has experienced. And I will say, I've experienced a lot of stress in the last 7 years. That was a lot worse than the initial stress that sent me into psychosis. So I'm happy to say that taking the Seroquel every day and going through like different dosages, you know, because having this disorder, taking the medication, the dosage will change based on different events that happen in my life. And so staying on the Seroquel has kept me from losing my mind again, you know. I deal with the depression by trying to rest as much as possible. And with the anxiety, I am on a medication for anxiety because it's just there, you know. So yeah, those are my main symptoms, the depression and anxiety.
Wow. Well, thank you for sharing all of that. And I just want to applaud you for fighting through all of that, because I think,
Yes.
as you pointed out, it's really difficult.
It's so challenging. Like, it was easier to go to school and get my degrees than try to keep myself balanced within the dysregulations, because what it does is I outside of taking the Seroquel, I experienced the dysregulation of sleep cycles, dream cycles, appetite cycles, energy cycles. That's a lot, that's a lot to deal with. But overall, I will say that I'm doing very well.
That's great. That's great to hear. And what I think is interesting too, as you pointed out, that you were told that it was a mood disorder, but all the symptoms that you're experiencing are definitely physiological. And it's not like you're really happy one day and then you're really sad the next day and you're sitting there thinking, why am I unhappy? You
Right.
literally cannot sleep, or you literally have no energy, and there's
no energy.
No explanation for it based on, your feelings. It's not like you're feeling sad, therefore you have low energy. You just have low energy.
You have low energy. And then the other marker is just the sensitivity of the five senses. So I smell things a little bit more. When I'm touched, I'm a little bit more sensitive, and that creates a little bit, it can create overstimulation, and I get depressed because of that, because it drains it. It's just, this is bipolar. That's how I would describe bipolar.
Yeah, it's a whole lot of things moving in very different ways and very different times. And I also want to touch on the insomnia and the sleep dysregulation, because I think for a lot of people, if I get a bad night's sleep, the next day I'm functional, but I can tell that I'm not functioning at 100%. But for someone who, as you pointed out, you weren't able to get a good night's sleep for months, and I think that's enough to put anybody into psychosis.
Right.
So having the dysregulation on top of that, I can't imagine. That's got to be awful.
And then lucid dreaming is a part of it too. The lucid dreaming is the worst. I'll wake up sometimes and I'm like, I need to go to sleep somewhere else where I'm not lucid dreaming, so that's another thing that's very active with my brain, I have a whole nother life when I go to sleep. So prior to my diagnosis, dealing with that, and like I said, waking up 10 to 12 times a day, we get called mean sometimes. Like, you just mean, you're in a bad mood. Well, imagine you're not getting sleep, So the way I treat my bipolar, because the range, the symptoms, there's a huge range. And so each person's symptoms, it's the same set of symptoms, but it's varying degrees from person to person. So what I make sure to do is make sure I'm treating the dysregulation of sleep through the Seroquel. So I will never say to myself, I'm going to come off this medication one day. Like, when you're diagnosed with bipolar, we need medication. It doesn't make me weak or anything like that, but my brain is different. My brain requires the medication. I have to take my medication, because I don't want to end up in the psych institute again. I don't want to go through that again. I'm grateful that I've only gone through it one time. So treating the sleep, it makes the depression not as bad, the fatigue, because like I said, I'm in a type of depression right now, but it's not as bad because I'm getting the sleep.
Yeah, that's crucial. I have one last question for you, Chandra, and then we're going to hear from Jenny. Despite, as you said, I really like the way that you put it, which is just your brain works differently. And so you need to be on the medication to be able to function at a relatively normal human level. And despite how many people like yourself, who are pretty high profile people that we know have had bipolar, from celebrities to politicians, there still seems to be this stigma against it. And I would say that we're recognizing now that there is a stigma against many mental health disorders, but I think it's interesting that bipolar has that stigma against it, even though it's a disorder where we can clearly say there are physiological, this is a physiological disease that manifests in, mental dysregulation and behavior differences. And yet there are some people who are ashamed to admit that they have bipolar. And so I'm just wondering, have you experienced any of that in your life, and what are your thoughts on that? I'm sure you have many.
So as you first mentioned, I have a master's degree. I own my own business. I have a bachelor's degree. I've lived quite independently. So the stigma for me happens in this way, where I'm told, oh, no, you're fine. You're okay. You know, when I say that I'm tired, I'll get, you know, everybody is tired, you know, and I think the stigma is there is because when we don't take our medication, we could be we could be little monsters. And so I think, like, because I actually have a website, it's chipping away the stigma of mental health diagnosis, you know, and I think if we can come into communities and provide information on what bipolar is in the way that normal people can understand, that'll help deal with the stigma. But people, when they don't understand something, it's judged, you know. And so if it's complex for us as medical doctors, what about just the normal people, you know? And so I think seeing individuals like me and even more individuals like myself, that's one way to start chipping away at the you know, at the stigma. Because the thing is, when I went through psychosis, everybody around me was affected. Everyone. And so my daughter to this day, she's like, Mommy, people talk about you so bad. They don't care that you have bipolar. It's hard for people to move on when they've been hurt by someone who went through the experience. Like you said, the regret, that's a tough thing, you know? And so if someone is like at the center or in the middle of what you're going through, then that's where the stigma comes from. So we just need some more people who are lights, you know, like, you know, we just need to see more light in the bipolar community.
That's beautiful. Thank you. So I'm now going to bring on Jenny, who is a colleague of mine at the Wyss Institute. Thank you, Chandra. And Jenny is working on a project called CircaVent that is aiming to bring more hope to people like Chandra and hopefully develop new treatments for bipolar. Because as we discovered, you know, Chandra's condition seems to be pretty well managed by one drug, but you know, one drug specifically for bipolar and then a couple of others, but many people don't have that luxury. So Jenny, you are the leader of the synthetic biology platform here at the Wyss. And can you explain what synthetic biology is for those of us that are not scientists, and how you and your team came to use synthetic biology to focus on bipolar?
Sure. It's nice to be here. And briefly, synthetic biology is a field that involves designing and engineering organisms to have new or new to nature abilities. And researchers can harness that power of nature to solve problems in medicine, agriculture, and sustainability. And at the Wyss, we have a very multidisciplinary team, and our researchers and faculty, including George Church, have pioneered new technologies, new transformative technologies to, for example, read, write, and edit RNA and DNA and proteins to create solutions for healthcare. And in particular to this, like one project that we had at the Wyss a few years ago, we published a technique to generate necessary cell types to build human brain organoids. So these are cerebral brain-like structures. They're not actual brains. And at the time, we were using these organoid models to study just genetic dysregulation in bipolar disorder in particular. And it has been a huge advancement in terms of understanding bipolar in like a 3D culture system from human cells, essentially.
So can you explain just briefly how an organoid is different from just growing some neurons in a dish? What makes that different and better?
So traditional like cell culture techniques, they're the cells are grown in 2D, so in a flat layer. And usually they're pretty one cell type. And with organoids, they're three-dimensional. So they have a volume. And with some of our technologies, we introduce, soon to hope to introduce more diverse heterogeneous cell populations, so we can study how cells interact in 3 dimensions, because as humans, we are in 3D, although in Zoom we are not right now. But typically we're in 3D to study how those cellular processes work together.
Cool. And so with these organoids, I think they're just made of neurons. So they don't have all of the structures and all the other cells that you would need to have a full human brain, but they do have some function. You know, they are alive in a dish. And so what are some of the limitations? Like what can you learn from, you know, a ball of 3D neurons in a dish, but what can you not learn from them?
Yes, so they are three-dimensional. They are, you know, considered like early development, but they're not what we consider like an in vivo model. So like a whole system beyond just the cerebral part here. Just 1 second. Okay. Never mind. And then, and at the Wyss, what we're trying to do actually from our CircaVent project, we just had the 3D human culture, but we really wanted to understand in vivo behavior. And as I mentioned before, at the Wyss, it's highly disciplinary. It's an amazing place to work, where we have engineers and they come from different backgrounds, for example. And I had a conversation just organically with a colleague, Richard Novak, who works with Don Ingber, and he and his team had developed a drug discovery algorithm to predict mostly FDA-approved repurposed drugs that could reverse the phenotypes for cognitive disorders. And they were working on different cognitive disorders at the time. And we talked about, oh, this is a very unmet need that affects a huge population of bipolar. So can we use some of your tools, combine it with our tools to really screen in a fast way to find drugs and drug treatments for bipolar?
And my favorite part of this is that it actually involves tadpoles.
Yes.
which I would not have thought would be a useful, you know, organism for studying bipolar, but it turns out that they actually are. So can you explain why tadpoles?
Yes, the tadpole system has been, to me, very revolutionary and extremely exciting for one reason. A lot of these studies to find drugs are often done in mammals, mice, for example. Mice take a long time to grow. They're incredibly expensive, and these studies can take a long time. So we wanted to speed up that process. So tadpoles are great in that you can create bipolar models very quickly, inexpensively, and use them in high numbers. So for example, our current system can screen over 1,000 tadpoles and look at their behaviors simultaneously. So we look at behavior, like swimming behaviors. We can do cognition and memory to look at, you know, how a model of bipolar affects a tadpole's in vivo, essentially.
So basically, if I'm getting this right, so CircaVent has 3 different pieces that you kind of put together. You have the human brain organoids, which are 3D balls of cells that have some function and that you can use to sort of mimic a human brain function, but, you know, not consciously. We're very aware of that. Then you have this algorithm that can predict what effects existing drugs might have. And then you have these tadpoles, which are basically a really efficient way to study the behavior of an entire organism when you subject it to different things. And so I think that's sort of the recipe, right, for coming up with bipolar drugs, because you need to have the human aspect, you need to have the whole organism effect, because as we learned, bipolar affects a lot of different systems in the body, and it has a lot of different, as Andy said, heterogeneous manifestations. But then you also have sort of the power of of machine learning that, as we know, is very good at recognizing patterns and parsing things out. So can you walk us through the linear process of how you actually use all these pieces of CircaVent to try to find new drugs for bipolar?
Sure. Yeah, we start with donated patient samples from both bipolar patients and healthy control patients. And we look at gene expression and how, like, across all of the samples, and we feed it into this drug discovery pipeline to essentially predict drugs that would reverse bipolar in a simplistic way. That's kind of what we're doing. And then we take our top hits. So we generate a lot of candidates. We take our most promising candidates, and then we screen them in our tadpoles. And as I said, the tadpoles, we have a large number, over 1,000, to look at this behavior. And then we screen again from there and put them and then screen in our organoids to see how they affect, like morphology. We look at neural function, firing, and then our hope is from there we have our top candidates that we would then move into, like, more into mouse models, for example, because getting it to that mouse model is actually very challenging, and we want to have the most promising hits out of that mouse models because those take so much time and they cost so much.
Wow, that is quite a process.
Yes, that is.
Quite a process, but what's interesting to me is, like, that's a whole process, but it's still faster than traditional drug development, which, as you pointed out, often takes place in mice. So is the main benefit just that the tadpoles are there, you can do a lot more of them, and is it kind of just a matter of numbers, sort of testing as often as you can, or is there something else about the tadpole system, I guess the whole system, right? The organoids, the tadpoles, the algorithm that makes your system faster, better than traditional drug development?
You've hit a lot of it. It's faster. It's you have higher numbers. The other thing about these tadpoles is, and what you don't see in mice either, is in mice, these are usually genetic knockouts that are knocked out in one particular gene. And although we knock out particular genes associated with bipolar, we do have a lot of genetic variation in those tadpoles, and that mimics closer to what you would see in a human population, because in bipolar and a lot of these diseases, there is a different genetic profile, so to speak. In addition, these tadpoles, although they're not mammals, we recognize that, they are, evolutionarily speaking, one step closer to other high throughput in vivo screening models, like zebrafish, for example. And so we're, trying to get as close to human as we can in a high throughput way. So the high throughput really helps us get those statistics to really see on a molecular level how these drugs could be promising candidates for bipolar patients.
Awesome. And I know that we heard from Chandra that there are a lot of different physiological manifestations of bipolar. And you mentioned that you're looking at behavior in these tadpoles. And as you pointed out, tadpoles are obviously not humans. Our behavior is very different. So what tadpole behaviors are you looking at that you're using kind of as a proxy for bipolar in humans?
So a lot of what we do, we look at swimming speed. So we see how fast the tadpoles are swimming. We see their swimming patterns. Another aspect of what we look at in our pipeline is circadian behavior. So one hallmark of bipolar that Chandra and Andy have talked on is a kind of like a sleep dysregulation. And so circadian with their day-night cycle disruption as well. So what we can't actually do in organoids so well that we can do in tadpoles is look at light cycle generation, like light cycle perturbations on tadpoles. So our system can put day, night, day or dark light cycles on our tadpoles to see how these light cycles modulate the bipolar phenotype. In addition to light dark regulation, we also want to look at dosing, timing of dosing, for example. So what we term a chronotherapeutic dosage to the tadpole. So can we modulate day night cycles, but in addition, like timing of the doses. So not just the drug itself, but is there a metabolic activity that's associated with bipolar in these tadpoles that will also reverse the phenotype? And we actually see in our tadpoles, in a bipolar tadpole, we do see, increased swimming behavior and a slight shift as to when these tadpoles start swimming. So we do have a bipolar phenotype, so to speak, that somewhat mimics some manic phases of our tadpoles, actually. So yeah.
Wow. I never would have thought that you could make a tadpole manic.
Yes.
I guess you have.
we have, and we have some promising hits too that have shown some amelioration as well in the tadpoles, and also we also see some bipolar phenotypes as well in our organoid models as well. So we can model bipolar, some aspects of bipolar, not fully, because I want to also recognize that bipolar is a mood disorder, is a psychiatric disorder, and we're trying to connect the molecular mechanisms of what we see for bipolar to the phenotypes and what Andy and Chandra have described, like in their own personal experience and clinically, in a clinical environment as well. So we do see some manifestations of that in our pipeline, and that will hopefully move toward, gives us like promising. Oh, sorry. Gives us like hopefully some promising therapeutic hits for bipolar.
Awesome. And what's the current status? So what is the next step now that you have these hits? Is it to, I think you mentioned maybe testing them in mammals, or, is there a point where we can hopefully see these in clinical trials? What's sort of like the next step?
So yes, so we are continuing to screen additional hits as well, and we're also looking at potential targets of what these drugs are targeting. And we are also, yes, we'd like to move towards the screening these in mammals as well, so in mouse models. So we're actively working on that front right now. And of course, we have this collaboration with Andy in order to connect some of what we're doing in the lab to what he sees in the clinic and how relevant some of our drug hits and how they could be potentially useful for his patients.
That's awesome. So I'm going to, thanks Jenny for lobbing me that softball. I'm going to bring Chandra back on screen, and we're going to talk a little bit about how the three of you have been sort of interacting and bringing your perspectives together to make this project, to move this project forward. So I know that Jenny, I think you were the one that reached out to Andy about getting involved with this. So can I ask Jenny and Andy sort of, well, let's start with Jenny. Why did you, why do you know that it was important, or why do you think that it was important to get Andy involved? Because what you're doing is really, for those of us that aren't in science, it's pretty hardcore, lab science bench work. And Andy is definitely on the clinical side. So what drove you to reach out?
Well, I think we knew in the lab how important bipolar is, and Andy and Chandra have talked about how bipolar has affected so many people. But again, we're in the lab and we don't really see these patients every day. I personally don't live it. And a big purpose of what we do at the Wyss, and I think what we all do as scientists, is we want to move our technology out into patients' hands. And the best way to do that is to work with, people like Andy, clinicians who see patients and understand what they go through, and also have an academic understanding of a lot of what we do, like on the bench work side. And so what we do in the lab is we do a lot of mechanistic molecular studies, but we don't really see the clinical side. And it's just been amazing to have you, Chandra, here to give a face to what we're doing and who we want to help. And so, I reached out to Andy and I said, hey, you're an expert in bipolar. And Andy has also worked with the Wyss before too. So he has an amazing track record. And so he's been phenomenal to work with, to give us direction and advice on as scientists, what are we doing that's relevant? And also having Chandra here has been amazing to have a patient perspective. What are we doing? Are we creating solutions that are helpful to you and that will benefit you? And that's why we're here. This is what all this work is for. So that's been beneficial to me, and how and why I reached out to Andy. So.
Oh, you're muted.
We lost you, Lindsay.
Sorry, I was muted. Can I ask Andy for your perspective and what's it been like, working with Jenny on this project?
So it's really been terrific, and I very much value collaborating with the Wyss Institute. One of the things I didn't really mention is that the last medication that was specifically designed for bipolar disorder was lithium in 1948. And since then, there haven't been specific drugs that have been developed. They were all taken from somewhere else. So I think to have an innovative platform to be able to identify potential new treatments, even if they're repurposed, which is much more efficient in many ways, that's great, because we just have to have new treatments. We have to get new treatments for better outcomes.
Awesome. And I also want to call out Chandra. I don't know if you want to show these, but I know that you have binders full of research that you've been doing, of tracking your own symptoms and your energy and your mood, which I think is just great. It really speaks to how patients can also be, involved in trying to help generate data to tell the scientists, oh my gosh, so many notes. I also wanted to reiterate what Jenny said, is that that you're living it, and you're also, you are helping to do the work that is going to be telling, people like Jenny, this is what my symptoms are, and really, we need a drug that solves X, or, my life would be so much better if this one particular thing could be taken care of. So just want to recognize you for that and all the hard work that you're doing on top of, dealing with bipolar itself. Do you have any final thoughts before we move into questions?
I'm just grateful for you guys. I'm grateful for Andy, because I am just a person that he didn't know. I learned about Andy through a documentary about a person who had bipolar and decided not to take their medication. And I'm like, let's see where this goes, And so I said to myself, here's this doctor from Harvard, I said, let's see if Harvard really cares about just the regular average person as compared to the celebrity that Andy was working with during the documentary that I watched. So I wrote to him and I'm like, look, I watched a documentary about bipolar and the person. Some of what you said, some of what medical scientists think is right, but here's what it really is. And so I was just hoping that he would talk to me. And so I'll say that you guys care, because here I am, just regular person who wrote in. And so, yeah, I'm just grateful to you guys for giving me the platform.
Awesome.
Thank you so much.
Thank you all. So we do have a few questions from the audience that have come in during the webinar. I'm going to start with a question for Andy. This is about bipolar diagnosis. I know that Chandra said that her diagnosis originally was anxiety, and I think and depression. And we mentioned how, a lot of the symptoms that can manifest are similar. So somebody wanted to know how bipolar is diagnosed, and how long does it take to diagnose someone? I mean, in Chandra's case, it took years and a psychotic episode before it finally was figured out. And what are the challenges of diagnosing bipolar versus something like depression that's very similar? And this particular person says that she is a woman with a hormonal disorder called PMDD, and apparently that is often misdiagnosed as bipolar because, like bipolar, the symptoms are cyclical. So how do you differentiate such a heterogeneous disease like bipolar from other things?
I'll try to be brief. But the hallmark of bipolar disorder is an unequivocal manic or hypomanic episode. And there are criteria that you can see that comes in a cluster, right? People have a need for less sleep and rapid speech and rapid thoughts and a loss of judgment and that sort of thing, all in a cluster at the same time. That being said, the challenge is that sometimes people are depressed first, and you cannot differentiate the depression of bipolar disorder from the depression of depression.
Right.
If somebody has not yet manifested a manic or hypomanic episode, you don't know if they will or they won't. And also sometimes I dare say that some of my colleagues are not systematic enough to carefully go through the differential diagnosis. PMDD, that's one thing where you can have some fluctuations. Mania is a completely different thing. So I think by being systematic, by looking at the criteria, and for anybody who's listening to contact the Depression Bipolar Support Alliance, because they can also be very supportive, where other people have been through it and may help people recognize whether they might or might not have the diagnosis.
Thank you. And there's a follow-up question, which is how early in a patient's life can bipolar be diagnosed? Because it's difficult to diagnose it in children because, their behavior is different than adults' behavior. So right now, how early can it be diagnosed? And is there a difference in how diagnosis goes for children versus adults, especially because it is hereditary, as you pointed out?
Right, and there are some very good offspring studies that show that really very young children, 4 or 5 years old, can have non-specific dysregulations. So you can have kids who can't sleep, for example, with insomnia, kids with anxiety, kids with school phobia, and that sort of thing. So what happens is that they develop a set of dysregulations and disorders over time until it finally manifests itself into a clear and unequivocal pattern.
Got it. Thank you. Jenny, we have some questions about the tadpoles, which I'm not surprised by. I feel like the tadpoles are the secret star of the show. One person is asking if the tadpoles have a fully functional blood-brain barrier. And I think the motivation for that was a secondary question, which is how do you create a tadpole bipolar model?
Okay, so I'll take the second question first. So for the tadpole bipolar model, there are genes that are have been associated with bipolar disorder. And what we do is, using what has been already published and discovered, we knock out those particular genes in our tadpoles. And I would say it's not a pure genetic knockout. It's what we call a mosaic. There's mosaicism where that there's a variation of that knocked out or knocked down in those tadpoles across our tadpole population. And so there are genes that are in literature that have been associated, and there are other genes that we have been looking at that have possible implications in bipolar as well. So we kind of look at it from two sides. So what has already been established and possibly new genetic targets for bipolar. And we use CRISPR to knock those genes out. And then we look at behavior of those tadpoles to see if they exhibit a increase, decrease in swimming behavior. We look at light cycle changes, and you could do like a memory cognition for those in order to say, what are these features in these tadpoles that have a correlation to bipolar disorder? So what you would see in a human patient. As to the fully functional blood-brain barrier question, we believe so, although we have not looked at it in particular. So these genes that we've knocked out have not been implicated in blood-brain barrier disruption, but we have not looked at that in particular. So.
Okay, thank you.
Yeah.
I have 2 more questions, I think is how we're going to be able to do in the 4 minutes that we have left. The next question is for Andy and Chandra. I'm going to open up to both of you. This question is about social factors and how they might affect how people manage their bipolar. For example, Chandra, you said that what triggered your psychotic episode was a very stressful event. And if people, don't have the support structures or, coping mechanisms in place to deal with stress, then that can trigger their bipolar or make it worse. And so this person is asking how that social aspect might impact treatment, and whether there is a clear indication of, if the treatment doesn't work well, like if there's a patient that does not respond to lithium, but they have, a very supportive social structure, will that person do well without medication? Or sort of how is that interplay? I'm sure that that's probably very complicated.
I will say for myself, I actually just took notes a couple of days ago, just wondering about the effects of bipolar on the 2 different personality types, the extrovert, introvert, where I tend to be introverted. So socially, that's a tough one for me because I have to have a time to myself to wind down and avoid the overstimulation that leads to the depression. And I'll say, as far as socially too, we kind of have to pick and choose like who and what we're going to be around. My situation that triggered the depression, it was work related. It wasn't something that I could avoid. But for me, socially, really just taking the time out to myself to balance social interactions, that's key to me. So for me, because like when I get to where I am now, I could be in a social setting and I'm not really involved, because of that introvert aspect. So I'll say the personalities has different personalities as it relates to different social events definitely impacts differently, Everything, you have to consider everything with this condition. Everything, socially, emotionally, physically, psychologically, all of those things have to be looked at, for me to be able to stay in balance.
I couldn't find it.
Great, thanks. And then I have one last question. Unfortunately, we're at our last minute of the webinar, but if your question was not answered, we will do our best to answer it via email, and we will send that email out to everybody who signed up for the webinar today. So the last question is for Jenny, which is, as a scientist who, as you said, you are in the lab all the time dealing with data and spreadsheets and graphs, how does having stories from patients like Chandra and having a connection with Chandra impact your work?
It's a great question because it's not something we have access to every day. And I think a lot of what, and I don't think I'm just speaking for myself, but for everybody who does scientific research, having that additional perspective is for one thing, very inspirational. I say this again, it reminds us of our purpose and what we want to do and our mission at the Wyss, and what we do as scientists, why we got into this to help people. Andy's lucky he gets to see people every day. I mean, I see people every day too, but in a different context. But it gives us an understanding that sometimes the data doesn't have. As we've discussed, this is a very poorly defined, disorder. We're still learning every day, and it's just that additional perspective of what are we actually seeing at the bench, and how does that translate in the real world to real people? Is this crazy swimming tadpole? Like, is that what we're actually seeing in patients? From some of Andy's stories, it kind of might be there. So yeah, it's been hugely helpful to have these additional perspectives.
Great, thank you. Well, we are exactly only 1 minute over time, which I think is great, given how much we covered today. So I just want to thank all of our panelists today, Andy, Jenny, and Chandra. Thank you so much for your time. And again, we will be emailing out a copy of this webinar. We're going to publish the recording to our YouTube channel. So if you have any friends who wanted to be here but couldn't, that will be available for them. And into the chat right now, we're going to put an email address. If you in the audience would like to learn more about CircaVent or learn about how you can support efforts like this one, please go ahead and email us. Someone does check that email address, I promise. And if you want to, if there's a way that we can work with you or that you'd like to support the Wyss and this effort, we would love to hear from you. So please don't hesitate to reach out, and check your inboxes in the next few days for those questions and for a link to the recording. And also that second link that just got put into the chat is a link to our CircaVent webpage that gives you a sort of a summary of how it works, if you want to send that to anybody that you know, and also has some really cool images, courtesy of Jenny and her team. So again, thank you everybody for attending. Thank you to our panelists, and I hope everybody has a great rest of your week.
Thanks so much.